Our research project is a data analysis of the foreign body response of cannulas being inserted into the eye. This project is by Angela Hernandez and Katriana Johnson. I'm currently helping with research in Dr. Basaglia's lab on ocular neuroengineering, and more specifically, the negative effects of pressure on the eye and the cells of the eye. To study this, a small cannula made out of silicone or a certain type of plastic is inserted into the eye and pressure is added to that. Cannula is just a small tube made out of plastic, silicone, or even metal that can be inserted into the body to add or take out fluid or gas. The problem that arose from these experiments is that at some point after the cannula is inserted into the eye, the tip of the cannula is completely encapsulated by fibrous cells. This causes a problem because it limits the amount of time that the test can be viable for, as eventually no more air pressure will be able to go through the cannula because it will be completely closed off. This is a type of foreign body response caused by the insertion of the cannula. According to the article by Arturo Vegas entitled Domesticating the Foreign Body Response, Recent Advances and Applications, the foreign body response is an immunological process that leads to the rejection of implanted devices and presents a fundamental challenge to their performance, durability, and therapeutic utility. The way we plan on solving this issue is by performing a data analysis and determining which materials or chemicals we can use to coat the tip of the cannula to prevent this fibrosis and encapsulation. The parameters with which we analyzed our data are the amount of drug that's released over time, the effectiveness on related devices, the effectiveness on tissue or related tissue, the side effects including possibly toxicity of some chemicals, and the target area that the original paper was based on.
The following is a list of the most current successful chemicals and materials we have found. Mitomycin C, also abbreviated as MMC, appeared in multiple of our found research articles, for it is effective in preventing fibrosis at the implant site. It covalently reacts with the DNA, forming cross-links between complementary DNA strands. This results in events such as mutagenesis and inhibition of DNA replication. In turn, it prohibits proliferation of fibroblast cells. Bifluorouracil is an anti-neoplastic agent deriving from uracil, which acts as an anti-metabolite. It also inhibits the synthesis of DNA by blocking the deoxyuridylic acid to diamidilic acid by cellular enzyme diamidylate synthase. It has many roles, but most importantly for this topic, it has a role as an immunosuppressive agent and anti-metabolite. Aperase and minocycline are both used to prevent a foreign body response on the cannula that's inserted into the brain. Aperase is an ATP hydrolyzing enzyme that hydrolyzes the extracellular nucleoside trisephosphates and diphosphates. By reducing extracellular ATP, aperase can suppress the extent of the foreign body response. ATP and ADP are also responsible for mediating chemotactic response of microglial cells that release pro-inflammatory cytokines. Minocycline is a semi-synthetic second-generation tetracycline with a broad range of anti-inflammatory properties. There is still further research that needs to be done on how it works exactly, but from what has been observed, minocycline was considerably more effective than aperase. Pirfenidone is a synthetic antifibrotic agent. It inhibits fibroblast, epidermal, platelet-derived, and transforming beta-1 growth factors, slowing cell proliferation. It also inhibits DNA synthesis and the production of mRNA for collagen types 1 and 3, resulting in a reduction of fibrosis. Polybeta homoserine consists of the hydrophilic unnatural amino acid beta homoserine. It can resist adhesion of cells, platelets, and fungi, all of which are widely encountered by implanted devices and may lead to a foreign body response. There is low fouling and low foreign body response towards this hydrogel as a result from strong dual hydrogen bonding hydration. OEG6 has strong repulsive force and is considered one of the best materials to resist biofouling. Simvastatin inhibits CXCR7, which is a gene expression responsible for inflammation. This allows for the production of M2 macrophages, which reduce inflammation. Some materials that were used to adhere the chemicals to the cannula were poly-2-hydroxyethyl methacrylate, also known as p-HEMA, and it allowed for the slow drug release of mitomycin C. In the presence of aqueous humor, the p-HEMA polymer swells, releasing the mitomycin C into the aqueous humor. Opal shale microparticles have a poriferous nature, a strong adsorbality, and are non-toxic. Thin films of polylactic co-glycolic acid, or PLGA, were used for the slow drug release of 5-FU and MMC. This material was chosen because it's biocompatible and non-toxic.
In our data analysis, we will be reviewing the most promising chemicals that we found, comparing the different chemicals, analyzing possible combinations of different chemicals, and then creating a conclusion where we will determine the best solutions based on the analysis, and then determine the following steps that we will do afterwards.
Mitomycin C was used in multiple articles to reduce the foreign body response. However, too much mitomycin C could cause serious side effects, such as corneal toxicity, lab leaks, or ocular hypotony. These figures show the release of mitomycin C over time. The top and bottom middle are the cumulative releases over time. As you can see, the dosage is very small. The bottom left and middle figures were used in combination with opal shale microparticles, or OSMP. The drug release was over a span of about 20 days. The OSMP structure allowed for a longer drug release. These figures show the percent of cells killed over time using mitomycin C. For this instance, mitomycin C was released from the surface of a single-layer porous PLGA film. You can see in panel F that the percent of cells killed significantly increases as the dosage of mitomycin C increases.
PLGA is another option for a coating for the cannula. In this specific example, PLGA microspheres were used in a combination with hydrogels that would allow the microspheres to disperse. This figure shows the cumulative percent of the drug released over time, which is very similar to many other drug releases, as it slowly accumulates before peaking at the top and staying pretty steady. PLGA is very adhesive to many materials and is also very commonly used in combination with other materials, as we will see later on.
These figures model a comparison of the viability and proliferation of paclitaxel and mitomycin C. The relative viability shown are from 4 various fibroblasts. Aparase and minocycline were both compared in this article. These figures show the performance of mice over time with the insertion of a cannula coated in either nothing, aparase, or minocycline. The minocycline, which is represented in black, has a better performance than aparase. For this experiment, 5-fluoroacyl and mitomycin C were both used to prevent foreign body response. For all the drug concentrations, the delayed release observed is because of the top PLGA sealing layer. These figures show the amount of cells killed over time using different concentrations of 5-fluoroacyl. There was an inconsistent toxic effect for the initial 5 days between 0.1 and 0.4 milligrams of 5-FU, and that was due to the sealed top layer with no drug content.
Unlike the other chemicals we've seen so far, polymethyl methacrylate, otherwise known as PMMA, is a chemical that is commonly found in implantations that was used in one article we found to induce the foreign body response. These figures show a comparison between the control and PMMA. In conclusion, the best solution that we've found is the use of mitomycin C in combination with another chemical to achieve the maximum longevity of the drug release, as well as to adhere mitomycin C to the outside of the cannula. The best combination chemicals that we've found are mitomycin C with FEMA, with PLGA, or with 5-FU with PLGA. Our next steps after this research project is in January I will start the preliminary test with the chosen material. I will start by testing the materials for their adhesion to the cannula by using old eyes that are not being used anymore. Then I will be testing the material's effect on the fibrous cells. And then finally I will start the testing of the chemicals on some of the prepared eyes.